In previous sessions, we have discussed about the carbohydrates. In one session, we discussed about the definition and functions of the carbohydrates. In the second session, we discussed about the classification of carbohydrates and monosaccharides. In this session, we'll be discussing about structural aspects of aldoses and ketoses. So, aldoses and ketoses can be explained either in a straight chain form or in cyclic form. Generally, they exist in the cyclic form when they are put into the solution. Okay, so first I will talk about the straight chain form. Okay, so that here I will discuss about the straight chain form. Okay, now let me see. So first I will take the example of glucose and fructose. They are exoses. First I will write the functional group. So here you say this is the aldehyde group and here it is a keto group. Then I will write primary alcohol group at the 6th carbon. Then I will write about the configuration around other carbons. Here it is hydroxyl group and here hydrogen and here again hydrogen and here it is hydroxyl group here. Okay. So hydroxyl group like second carbon, here also hydroxyl group and second carbon. Okay, here also there is CH2OH group and here this one, here there is a hydroxyl group and here right side hydroxyl and right side hydroxyl. It is very simple. So it is what we call D-glucose. It is D-glucose. What is D-glucose and what is L-glucose? So I will explain when we talk about the isomerism. Okay, there are various types of isomers are present. These are a straight chain structure. It is a straight chain structure. Okay, then. So as I said, when you put into this one, so they don't remain in this format. So they attain when you put in the solution, so when you dissolve them in solution, they attain a structure called hemiacetal. Hemiacetal in case of aldoses, in case of ketoses, that is hemiketal. Okay, so there is a internal cyclic structure form takes place. How this is forming? So let me explain. It is very simple to understand. Very simple. Okay, let us see how this cyclic structure looks like. Again I have to write exose. Okay, so this is exose with 6 carbons. Same thing which I followed earlier. Here again a fructose with 6 carbons. Okay, there are 6 carbons. Then what I do is now in this case, now I will write a primary alcohol group as there is no change in the primary alcohol group. Okay, here what happens, this is the first carbon, second, third, fourth, fifth and sixth. Like also first, second, third, fourth, fifth and sixth. So now see how this cyclic structure form hemiacetal form is taking place. See here. This is the first and fifth carbon. First and fifth carbon. Then you can see the configuration around other carbons. Here OH on the right side, hydroxyl group, and here you see if you can refer back to the straight chain, it is on the left side. Again here, okay, then here it is H. Here again, here it is between second carbon, okay, here between second carbon and then fifth carbon, okay, then it is, okay, then it is like this, okay, then here it is, okay, then here it is. See how beautifully the structure form has taken place. Okay, so what we say this one? We say alpha D glucopyranose. Alpha D glucopyranose. Here it is alpha D fructofuranose. So when I say alpha and then beta, so then there may be a beta also. So that also I will try to explain. So pyranose comes from the pyron and puranose comes from the furan. Okay, that is the chemical structure. Now let us see how they can be represented. So they say they Haworth projections. Haworth projections, alpha and beta can exist. Let us see here. Alpha and beta can exist. Okay, this is the one. Then next, very simple, very simple. Okay, then here it is. Here it is. Okay, this is the first carbon. This is the second. This is the third and fourth. This is fifth, and here also this is the sixth one. Okay, here it is first, second, third, and fourth, fifth, and you can say it is sixth. Then I will write the configuration from starting from the second. OH, OH. Here it is reverse here, you can see, and here we say OH here below the plate, and here it is, okay, like this. Here also second carbon, no change, OH here, and here it is H, OH, okay, OH, and then H. Here it is CH2OH. Now let me see the first carbon. The first carbon, okay, here let me see the first carbon. When I talk about the first carbon, what is the configuration around this one and this one? Here, see the change in the configuration. Here OH and here H, and here it is H and it is the OH. So just to make it more clear to you, so here you can see, observe the configuration around these things. Also observe the configuration around this one. Okay, so just observe it here. So this is the below the plane of the ring, and it is the above the plane of the ring. The distance you can see, it is a much far distance, it is near distance. Okay, when you see both are in the same plane, CH2OH and OH, here it is not like that. So here what we say is this one is alpha D glucopyranose. Alpha D glucopyranose. It is, we can say, beta D glucopyranose. So pyranose. So you can say, why you are explaining these structures? What is the importance? So my friends, it is very important. You know the enzymes. Enzymes are highly specific. Some enzymes, they can act on alpha D glucose. Some enzymes, they act on the beta D glucose. So there is enzymes, they are minutely selected. So they have a microscopic vision on which molecule they have to act. I will write the furanose structure. So now again I will write this one. Okay, here. Okay, so like this, here it is, and here it is. Okay, then here it is. Okay, then I am writing again here. Okay, then it is furanose structure, because second carbon the structure is there. Here it is what it is. Here this is the first carbon. This is the first one. Okay, that is the one which is taking place. Or then, so what we say is this is the first carbon. So this is the second carbon. So this is third carbon. This is fourth carbon. This is the fifth carbon. Okay, this is the fifth carbon, and this is the sixth carbon. This is the sixth carbon. So like also this is the first carbon, second, okay, third, fourth, okay, fifth, and sixth. So that is slightly different from the, so that is pyranose structure. Here what happens here? Here it is CH2OH. That is CH2OH. Okay. Then here it is the first carbon. Okay. Then here you can say CH2OH. Here H. Here H. Okay. So sorry, OH. OH. Here OH. Okay. Then here it is H. OH. Okay. Here it is. Then here it is. Here like that. Okay, here you can say it is again H, OH, OH, okay, H, okay, CH2, OH. It is called alpha D fructofuranose and beta D fructofuranose. Okay, this is about the furanoses and then pyranoses. They attain, pyranoses and furanoses, they can attain a boat shape or chair shape. But both shape are very rare and generally they exist in the assume the chair shape. Okay. So this is about the various structural aspects. Now I will summarize. Whatever we have studied, I will summarize. Okay. Then in this one, in this session, so we have discussed about the structural aspects. The objective was at the end of the session, the learner shall be able to describe structural aspects of aldoses and ketoses. First, we discussed about straight chain. Okay. Then, in the straight chain, we have taken the example of glucose, hexose sugar, how the configuration looks like. Then they don't remain like that when you put in the solution. So there is attains the cyclic form, that is acetal or ketol. Then we have shown these various the structure and the that is in glucose it is glucopyranose and in the whereas so we say this is fructofuranose like that. So it attains that structure. So then they attain the Howard projections. So our projections in the cyclic structure it is so represented either boat or chair form. So let us see how the terminology is given. If the in this case the OH is below the plane, that is alpha D glucopyranose. If OH is above the plane, that is beta D glucopyranose. Whereas in this one also we have shown this is the alpha D fructofuranose. It is so beta D fructofuranose. This is the way how the structures of glucose and fructose are written, but you don't have to write it, but you have to, for understanding it is must. Okay, in that way there will be clarity of the understanding. Okay, in the next session, so we'll talk about the various aspects of the isomerism. Okay, thank you very much.